Julian Doe and Shireen Davis. There he studied calcium signaling in the renal tubes of Drosophila. Later on he came here to Cambridge to do a postdoc with Professor Andrea Brand. There he investigated molecular mechanisms that control neural stem cell differentiation. This work included the identification of a novel tumor suppressor and also the development of an in vivo profile technique that he's going to talk about today. That work garnered him a position in the Imperial College of London, where he is right now setting an independent lab. So without further ado, here is Tony.
Thank you very much, Monica. It's an honor and a pleasure to be able to talk to you today about my research. And I'd like to thank Monica and Paul for arranging this today. So, since I started my postdoc, I've been interested in how protein DNA interactions regulate gene expression. So next slide, please. So why do we want to know where proteins bind in the genome? Well, the way that a cell develops, the fate it obtains, how it reacts to its environment, or whether it forms cancer or not, all depends on gene expression levels, and these are controlled by interactions between proteins and the DNA. For instance, you have RNA polymerase, which unwinds the DNA and generates messenger RNA, and the DNA itself is wrapped around histones, which the histones in themselves can control expression levels through post-translational modifications. Also, another key thing is that transcription factors can bind to the DNA and influence gene expression, either increasing activating gene expression or by repressing it. So all of these things together need to be studied in detail if we're to understand the process of development and gene expression level control. Next slide, please. So also, you can imagine if we're interested in how a transcription factor regulates a certain process, we want to know where in the genome it binds, so we have an idea of what the genes are that it's regulating. And also, if we combine that with other techniques, we can start to pull apart these transcriptional networks, so we can work out whether the transcription factor directly binds a gene and turns it on, or whether it binds and turns on another transcription factor, which in turn turns the gene on. And so with the new technology being developed, we were able to start doing these kind of experiments. Next slide, please. So one of the most commonly used methods for looking at protein DNA interactions is chromatin immunoprecipitation, or CHIP. So this involves cross-linking your proteins to the DNA, either with formaldehyde or other forms of cross-linking. You then have to shear the DNA so that you have it in small pieces attached to proteins. And then you can use antibodies specific to your proteins of interest to pull down those proteins with the DNA attached. Once you've done that, you unlink the protein from the DNA, and you can then profile that DNA. So previously people used microarrays, but now they're using high throughput sequencing. Next slide, please. So for CHIP, the advantages are that you can get very high resolution. So this is really good for working out, if you're interested, the transcription factor binding motifs. You can also profile specific histone modifications. You can generate an antibody against a specific modification on the histone and then pull down those histones that have that modification. And another advantage compared to the next technique I'm going to tell you about, it doesn't require the generation of a fusion protein or to make a transgenic organism. However, there are some disadvantages. One of the main ones being that you require a specific antibody. And for some proteins, it's very hard to get an antibody that's specific enough, or at all, to do these experiments well. And often each experiment, depending on the antibody and the protein, will require some optimization. And Also, depending on the experiment, what kind of cells, what kind of protein you're profiling, you can need a lot of starting material, which can be a problem for certain investigations. Next slide, please. So, back quite a long time ago now, when I started my postdoc, we were interested in a protein which I'm going to tell you a bit about in a minute, but we could not get an antibody to work to do chip experiments. So at that point, we decided we'd try an alternative technique called DamID. So this is DNA adenine methyltransferase identification. So what we do with this is we take our protein of interest and we fuse an adenine methyltransferase to it. We then express this fusion protein at very low levels, either in our cells or in a transgenic organism. Then when the protein interacts with the DNA, it leaves a methylation mark. So the methylates adenines in the GATC sequence. So throughout the genome, wherever that protein is bound, you get this signature left in the DNA. And we can isolate those sequences to profile. And this was originally developed by Bas van Steensel and Steve Henikoff in 2000, 2001. We've used it quite extensively over the last few years to investigate neural development. Next slide, please. So hopefully you can see here a schematic of how we process the DamID material. So every experiment we do a control experiment which has a DAM only, so just a DAM on its own to control for background methylation. So once we've done the experiment, the DAM has been expressed in the tissue for a certain amount of time. You can freeze the tissue or cells, and then you just do a standard genomic DNA extraction, and then you cut the DNA with a restriction enzyme called DPN1. And DPN1 only cuts the methylated GATC site. So you can see those in orange. And so once you've treated DPN1, they're cut out, and then we use ligation-mediated PCR to amplify those fragments. And what we used to do is we then have our experimental sample and our control sample, and we would label one red, one green, and hybridize those to microarrays that tile the genome. So that was the original way of doing it. Next slide, please. So advantages for DamID, obviously we don't need to make an antibody. It's all DNA based, so we don't have to work with unstable proteins or RNA, so we don't have to go in the cold room, which is always a benefit. There's high reproducibility between the biological replicates because it's expressed quite well. You have accumulation of methylation. The profiles we get out are very similar, and also it doesn't require much starting material. However, the disadvantages are it's limited by the resolution is limited by the frequency of the GATC sites in the genome. They should be about every 250 base pairs, but obviously in different parts of the genome that can vary wildly. Another thing it can't do is profile histone modifications. However, we can profile the enzymes that either write or read those histone modifications. Another disadvantage of the original system is that there's no spatial temporal control. So we were expressing the DAM fusion protein all the time in all cell types. Next slide, please. So what I'm going to do is just tell you a little bit about the system we were studying originally. So I was interested in Andrea's lab in the mechanisms that control neural stem cell differentiation in Drosophila. So the neuroblasts in Drosophila undergo asymmetric divisions. They produce another copy of themselves, they self-renew, but they also create a smaller daughter cell called the GMC. The GMC then only normally divides once more to give neurons or glia. And during these asymmetric divisions, there are certain proteins held at the cortex of the neural stem cell, so at the apical and basal cortex. And if we go to the next slide, sorry, we talked about that. And the next slide, please. So you should be able to see here a picture of a confocal image of a neuroblast, showing the DNA in blue, and a protein called Prosper in red. So Prosper is a protein that's held at the basal cortex of the the neural stem cell. When this asymmetric division occurs, phospho is segregated to the daughter cell, where it can enter the nucleus and act upon its transcriptional targets. So it's an atypical homeodomain transcription factor. And obviously, we were quite interested in what the genes are that phospho regulates. So it had already been implied that this was a key factor in the switch from a self-renewing state to a differentiating state. So we tried the DamID system. We fused phospho to DAM, and we made transgenic Drosophila that expressed this fusion protein at very low levels throughout the organism. Next slide, please. So hopefully you're on a slide here where you can see an example of the data we got back from doing this experiment. So here, phospho is binding to the cyclin E gene, and we can see that we have here a ratio, a log2 ratio between your experimental sample and a DAM-only sample. And we see that there's a high ratio for experimental DAM horse sample in the intronic regions of cyclin E, which is promotes cell cycle. So we were very excited when we got this data, and we had the data for the whole genome. And taking that data, we then did another experiment where we took mutant phospho embryos and looked at the transcriptional state of genes and compared it to wild-type embryos to see how these genes that were being bound were regulated. Are they being activated, or are they being repressed? So to cut a long story short, if you go to the next slide, we found that phospho was indeed acting as a binary switch between the two cell types. So it would enter the nucleus of GMC and directly bind and repress neural stem cell genes. But we also found it bound to a lot of differentiation genes that haven't come on yet, suggesting maybe it was priming these genes to come on later in development. And so that was work I did with a PhD student, Sunil Choksi, we published a while ago. So we move to the next slide. So, as I said, the DamID technique was very useful, but we had that main disadvantage that we couldn't look in specific cell types, and we couldn't look at specific temporal windows. So obviously, organisms are comprised of many different cell types with very different properties. And you can see on the right of the slide a beautiful picture from the rainbow technique. So those are all different neurons in the mouse brain that are labeled with fluorescent proteins at different levels. And so in the brain, obviously, all these different neurons have different properties, like the neurotransmitters they produce, and the electrical properties, the preference for synaptic targets. So if we want to start to understand how these neurons are formed and how their properties are maintained, we need to be able to profile what's going in those specific cell types. And it's been quite a hard thing to do up until recently, as you have to, most of the time, you have to separate those cells from the rest of the tissue. So what I began to do many years ago was to try and adapt the DamID technique so we could use it in a cell type specific way. So we could just label the DNA in the cells we're interested in, and that way then we could extract the DNA and profile those specific cell types. So next slide, please. So sorry, this should have been a movie, but because of the technical complications, I can't show you. But this would have been a movie of just adult Drosophila brains, as my student took over the summer. And then all these little dots in the brain are cholinergic cells. So we can label all the, specifically only the cholinergic cells in the Drosophila brain. And I'll come back to that later because we've been starting to profile those different cells. Okay, next slide, please. So before I continue, I'll have to tell you a bit more about Drosophila genetics. So Drosophila is it's been a very useful model organism, and one of the most useful tools that's been used is the UAS GAL4 system, which was actually developed by my old boss, Professor Andrew Brand. So the idea is that you have GAL4, which is from yeast, which binds to UAS sequences and activates gene expression. And so many different Drosophila lines have been made that have GAL4 expressed in different cell types. If you take that line with GAL4 in it and cross it to another Drosophila line which has UAS sites and another gene, then you can activate the expression of that gene to stop it. So you could cross it to a line that has UAS sites and GFP, and then those cells will be labelled fluorescent. So we have lots of these different GAL4 lines, and we can create our own UAS lines. Next slide, please. So, so the traditional DamID system was used by just having our UAS sites and the DAM fusion protein. And we didn't add any GAL4 at all, and it was actually low-level transcription from that UAS promoter, HSP70 promoter, that gave just a tiny amount of RNA and a tiny amount of protein, and that tiny amount of protein was sufficient to give us a signature. So the DAM transferase is very active, and you just need a small amount. However, if you add GAL4 into the system, make a lot of RNA, make a lot of protein, you saturate the methylation in the genome, and it's actually very toxic. And when we did this, the embryos would arrest and die. So, in its current state, we couldn't use it to do cell type specific profiling. One, because it was toxic, and two, because it would still be expressed everywhere. So the idea I had was, next slide, please. So are you okay? So hopefully you're on a slide that says targeted DamID at the top. Yeah? Red? Yeah? Okay. So the idea I had was if we could maybe reduce the translation of the mRNA that's produced, then we could reduce the amount of protein that's made. So there's this phenomenon called ribosome reinitiation, where if you take a reading frame and put it upstream of your reading frame of interest, sometimes the ribosome will skip and start the second reading frame. This is without an iRes, it's just 2 stop codons and 1 base. But the frequency at which it reinitiates depends on that primary reading frame. So what I did is try different sizes of reading frames. So the small reading frame still gave us lots of translation of the second reading frame, the DAM fusion protein. But I kept increasing it until we got to a point where the levels were low enough that it wasn't toxic, and also that there wasn't any protein expressed in cells with no GAL4. So it ended up being actually a primary reading frame of M-cherry, which was quite handy because it also labelled the cells red. Next slide, please. And so here's just one of the figures from the paper. So if we amplify methylated DNA from the Drosophila genome from wild type, and from fly, the DamID flies with no GAL4, we hardly see any products. But when we add GAL4 into the system, we get methylated DNA. So that was very encouraging. Next slide, please. And so we have now modified the DamID to allow cell type specific profiling and protein DNA interactions. What's great about it is we don't have to isolate the cells or nuclei to do this. So we can just freeze the tissue or even the whole organism, extract the DNA and carry on. There's no immunoprecipitation, so we don't need any specific antibodies. It's highly specific, so we only get a signal from the cells that are expressing the protein. And I've actually been able to profile 100 neurons in a whole adult head, which is about maybe 200,000 cells, and still get a good signal. It's highly sensitive, as I said, and also we can control when the GAL4 is active using a temperature-sensitive GAL8. And so it's an adaptable technique, as was the original technique. We can look at transcription factors and chromatin remodeling enzymes. But what's been really, really useful is to look at RNA polymerase II. So go to the next slide, please. So obviously, if we could profile where RNA polymerase II is interacting with DNA, we could assay the transcriptional state of specific cells in vivo in a transgenic organism very easily. So I ended up using the core POL2 subunit, about 215 kilodaltons. Next slide, please. And so this appears to work fine, which is quite surprising considering that the polymerase machine is actually unwinding the DNA as it's transcribing. And the methylase must be methylating the double-stranded DNA either side of this process. But however, it still works, and we get similar profiles if we compare it with a POL2 chip. Next slide, please. I'm going to tell you very briefly the system we tested the technique in. So now I've been telling you about Prosper, and that work was done in the embryo. But now we're looking at the larval brain of Drosophila. So the life cycle, Drosophila, you have an embryo, hatches into a larvae, goes through 3 different stages, then it pupates and then turns into an adult. So this is in the last stage of the larval phase. And in the brain, it has 2 populations of neural stem cells. One that's symmetrically dividing, and then one that's asymmetrically dividing. And actually, the symmetrically dividing ones gradually change into the asymmetrically dividing ones. You can see on the arrows on the diagram. So what we wanted to do was to profile RNA POL2 binding in these 2 different populations and compare them. We already knew some differences between these cell populations, so it would help us prove our technique, and it also might give us some new information. Next slide, please. So here, hopefully, you can see some examples of the POL2 binding plots for a couple of different genes. So at the top, we've got POL2 binding at the bearded genes Tom and bearded, which involves in notch signaling. And you can see that in the new epithelium, the symmetrically dividing cells, we have POL2 binding throughout the gene structure, but in the asymmetrically dividing cells, it's only at the 5' end. It's as if the POL2 is stalled. And when we look at the RNA, you can see the in situ on the right, that this is Tom RNA is only expressed in the cell types where we see the POL2 binding across the gene structure. And similarly, for the JAK-STAT ligand unpaired, we see binding in the neuroepithelial cells, but not in the neuroblasts. And when we look at a trap line, we can see that it's only expressed in the epithelium. So it seems to be working well. Next slide, please. So that was the work we published in 2013. But as with the original DamID technique, this targeted system could be adapted for other organisms. Obviously not yeast, but plants. It could be done. C. elegans is another one, and my old lab are trying to adapt it to be used in mice. And hopefully, once people start using it and realizing how good it is, it'll be a powerful technique. Next slide, please. So at the end of the talk, I'm just going to tell you what I'm doing now as I'm setting up my new research lab. So I'm interested in how different neuronal properties are specified and maintained. So as I mentioned before, there's probably about 80 billion neurons in a human adult brain with very diverse properties, neurotransmitters, electrical properties, etc. So how are these properties specified and maintained? And also, what are the most appropriate factors reprogramming neuronal fate in vivo towards a possible therapeutic use for the technology to reprogram neurons for nervous system repair. So obviously I've been using targeted DamID. Next slide, please. So hopefully you can see a flow-through chart here for experimental protocol. So we can use in Drosophila these different GAP4 lines expressed in different neuronal populations. So initially I'm looking at neurotransmitters. And so I'm profiling RNA POL2 binding in these different cell types and comparing them to see what's different. And also be using yeast hybrid screens to complement that. And after we get all this data back, we can prioritize these factors and see what their role is in setting up neuronal properties during development, but also whether we can use these factors to reprogram neuronal fate. Next slide, please. So here's some data we've got quite recently. If we take cholinergic neurons, so we've got this char-gal4, which only expresses in those neurons. We've crossed it to the DAM POL2 flies, and I've taken adult heads. So this is quite easy because if you take about 30 adult flies, you freeze them in liquid nitrogen, and if you then shake them vigorously and put them through a sieve, the heads will fall through the holes and you can collect them very easily. Sounds a bit macabre, but it's very quick. And so we've done that for cholinergic, glutamatergic, and GABAergic neurons. And so if you look here, this was very encouraging, because here we can see that the transcription factor ACJ6 is the only factor that's known to promote cholinergic specification in Drosophila, and we see that that is highly bound by RNA pol 2 in the cholinergic neurons, but not in the other cells. So that was very promising. Next slide, please. And here's some other uncharacterized genes that seem specific. Here's another one that's specific to cholinergic neurons. Next slide, please. And we have an ETS transcription factor which is expressed in glutamatergic, GABAergic, but not in cholinergic. So that's quite interesting, and we'll probably be testing whether that factor inhibits the cholinergic fate. And then another homeobox transcription factor which is enriched in glutamatergic. Next slide, please. So what I want to do eventually is to use the Drosophila brain as a model for reprogramming the brain to repair itself. So something I haven't told you about is I'm also interested in maybe how we can de-differentiate neurons to create a pool of stem cells. So imagine if we do that in the soft adult brain, we create these pools of stem cells, and then from what we learn about how to specify neurons, we could then generate new neurons in the brain. First of all, can we do that? Second of all, do these neurons function? Do they integrate with existing circuits? I think this would be quite exciting experiments to do. And luckily, Drosophila has a lot of genetic tools, so I think it can be done. Next slide, please. So, summary for DamID. DamID can be used to profile protein-DNA interactions in vivo. We've adapted it so that it can be used in a cell-type-specific manner. And we've used it with pol2, which has been really, really useful to look at the transcriptional state of cells. And it's my previous lab have also looked at transcription factors, chromatin-modifying enzymes. So it's very versatile. And hopefully I've persuaded you that this is a method of choice for cell-type-specific profiling. So it's simple, no cell isolation, no antibodies, sensitive and highly specific. Next slide, please. So with that, I would like to thank people. So most of this work was done in my postdoc lab, Professor Andrew Brands, and I have lots of help with the target DamID from Boris, Kat, Owen, Libby, and Catherine. And also I'd like to thank funders and FlyChip. And I'd also like to thank Monica and Paul again for inviting me for this talk. Thank you very much. Any questions? Mm-hmm.
Is it possible to use this method to identify transcription factor binding sites which are far away from the transcription start site? Because most of the data you're showing, because it's with RNA polymerase II binding, it's in the exonic and intronic regions of the gene. How about far upstream sites? Is it useful for that?
Yes.
Critical transcription factors that might be involved in the regulation of those genes that you're looking at.
Yes, so the data we get back is for the whole genome, so we can see where transcription factors bind, not, as you say, just near the gene, but further away. Obviously, it gets a bit tricky when there's lots of genes, because it's hard to tell whether the transcription factor binding is regulating the gene right next to it, or maybe it's regulating the one over it from it. And I think that's a problem for any technique looking at the transcription factor binding.
And apart from looking at RNA polymerase II binding,
yeah,
that specific transcription factor, I mean, you showed some target genes, but other transcription factors besides RNA pol2?
Yes, we looked at Notch, so we took the intracellular domain of Notch and looked in those two different cell types, so the symmetrically and asymmetrically dividing stem cells, because it's known to have a role in the symmetrically dividing cells. And we got very good data for that, and we've looked also, people in my lab have looked at phosphor in a cell type specific manner, and other transcription factors related to phospho function. So far, we haven't had a failure when looking at any transcription factor. So it seems to work. I guess one thing you have to consider is where you tag the transcription factor with the DAM. So normally, either we've tagged it with GFP4 and we know it's okay, or we try and put DAM at the end of the protein, which is furthest away from the DNA binding domain.
It doesn't hamper then the interactions that might be happening between the transcription factor, let's say C-terminal, with other transcription factors, having the DAM there.
It is possible. We see strong binding. It could be feasible that some of that binding, we don't see some binding because it's not interacting. That could be possible. But it's quite hard to rule out unless you also do a chip experiment to confirm it's the same.
Thank you so much for your excellent seminar.
No problem. Thank you very much.
Thanks everybody for receiving us. We're looking forward in working with you, bringing you more seminars that you might be interested in. So if you guys think of another topic that you guys want us to bring to you, please let us know. We have our website, which is Cambiowebinars.wix.com/home. Also, I think Dr. Carmen is going to pick up the attendance so that we can send you a link to get feedback from you, and we can continue tailoring these talks to continue bringing science to Puerto Rico. Thank you very much.
Thank you.